Hello everyone. Today we are going to present our final project on developmental trajectory analysis of single cells in the human embryonic prefrontal cortex. Here is the content of our presentation. We'll start by introducing the human prefrontal cortex and the dataset used, followed by 3 parts of our project. The human prefrontal cortex is located in the frontal lobe of our brain, as you can see in the illustration. It plays a crucial role in higher-order cognitive functions, such as decision-making, working memory, and social behavior. Single-cell sequencing of human PFC cells at different gestational weeks enables the investigation of cell subgroups and developmental trajectories. There are different strategies for trajectory analysis. For example, the figure in the middle shows the results of modeling gene expression as a smooth function of pseudotime along lineages, and the right figure shows a topology-based computational analysis to study the differentiation trajectory. In this project, we aim to identify subgroups of cells of the human PFC and to trace the developmental trajectory using single-cell sequencing data. To achieve this, we utilized a published dataset containing 2,394 single cells from gestational weeks 8 to 26, as you can see in the figure. The number of cells for each developmental stage is summarized in the table. The expression data matrix integrates 24,153 genes for each cell in TPM. In the first part, we identified 9 different subgroups of cells through hierarchical clustering using the CRAD packages, as you can see in Figure 1a. The cell subgroups were distinguished according to curated cell-specific expressed marker genes, and Figure 1b shows the expression of a list of marker genes in different cell clusters. As you can see in Figure 1c, there is a total of 9 cell subgroups identified. We then annotated the cell clusters shown in Figure 1d. Since some cell clusters shared the same set of marker genes, we grouped them into 1 cell types. For example, clusters 0, 3, 4, and 6 were annotated as excitatory neurons.
Now I'm going to introduce 3 common trajectory analysis methods and select one that has the best performance. Molecule is the widely used trajectory inference methods designed for single-cell transcriptomics data in use an algorithm that takes single-cell RNA sequence data and arranges cells in pseudotime, providing a high-resolution view of cell developmental or differential expression, allowing user to find genes that change the function of progress through the biological process. Slingshot is another trajectory inference method. This method is designed to infer continuous branching linear structure in low-dimensional data. It uses cluster-based minimum spanning trees to initialize pseudotime trajectory and feeds simultaneously principal curve to capture the common trends across the lineage. And the major advantage of the slingshot is its ability to handle complex branching trajectories which can occur during the process, like cell differentiation. Lastly, T-scan also first cluster cells and then construct the minimum spanning trees among the cluster, estimate the number of the different cell states and arrange them in pseudotime to model the transition of cells from one state to another. Our primary object was to determine the most accurate and reliable method for reconstructing cellular developmental trajectories in our data. To evaluate the performance of each method, we first calculate the Pearson correlation between the UMAP1 dimension and inferred pseudotime values and the week time, which is our ground truth. According to Figure A, our finding indicates that UMAP1 dimension is inversely correlated with the week time, suggests a negative relation between the two. However, molecule exhibit a strong correlation with the week time, indicating that its inferred trajectory closely aligned with the ground truth timeline. This suggests that molecule can accurately capture the temporal dynamics in the data. We draw the molecule trajectory in the UMAP plots, which is shown in the Figure B. The plot validates our conclusion that trajectory is correlated with the week time, but inversely correlated with the UMAP1. On the other hand, slideshow shot performed the worst among 3 methods. It did not correlate with the UMAP1, but also or the week time, suggests that its inferred trajectory significantly deviates from both the underlying data structure and the ground truth timeline. And based on this result, we can confidently concludes that molecule is the most accurate and robust methods for trajectory analyzed in single-cell RNA sequence data among the ones we evaluate. Therefore, in the following analysis, we regard molecule as the preferred methods for reconstructing cellular developmental trajectory.
Then let's take a detailed look of the trajectory inferred by the molecule 3. We can see that the overall trend is aligned with the ground truth time labels. Along this arrow, the time migrates from week 8 to week 19 and to 26, which is well aligned with the node numbers in the trajectory. Also, we can see that the excitatory neuron further evolves into several subtypes, which is also reported in this paper. To further investigate the performance difference, we look into the temporally varying genes. Specifically, we use generalized additive model since it can model nonlinear relationship really well and figure out the most important variables. Therefore, we build a model for every gene expression versus the pseudo time. Then we select the top 2 genes for further visualization. Here shows the gene expression change versus the first component of the UMAP embeddings, and the cell types are labeled by different colors. Recall that the molecule has a strong correlation with the ground truth. T-scan has some median correlation, while slingshot has very poor correlation. In these figures, we can roughly identify blue, orange, and brown arrows, which are excitatory neurons, interneurons, and microglia cells. In addition, astrocytes are mixed with other cells but can be better separated in T-scan. However, there are significant differences in NPCs. We can see that they are clearly separated in monocoles, partially mixed with neurons in T-scans, and can be hardly identified in slingshots. Despite that astrocytes can be better identified in T-scans, it seems that correctly separating NPCs is more beneficial. The reason may be NPCs are root for all cells, and correctly finding those stem cells may be more important in trajectory inference.
Okay, this is the third part of the topological and data analysis, and for the whole process, the work are done with the Python package sctda. The first step after we transfer data into the longitudinal format is to data preprocessing about the filtering some cells and also get some important genes. After subsample the cells to pass all the criteria, we model the dependence of the dropout rate on the average gene expression by fitting a fitting function. And after the function is fitted, we use the above model to identify genes that have a low probability of dropout and have a higher variability than expected from purely technical noise. And we identify these genes as the high-quality genes, and totally 86 genes were selected for further analysis work. Below is the topological representation of our data. The left one is a simple dimensional reduction by PCA and its representation, and the left part is the topological representation. And the top one is the unrooted representation of the data, and the bottom one is the rooted representation of our data. And these two are all calculated by the algorithm as map. After the topological representation is constructed, we would like to try to reveal the developmental trajectory of the PXDs based on these topological data. So for figure A, we visualize the rooted topological representation based on a time point. The red color means the late time, and the blue color means the early time. And we can see from figure B is the spectrum of the network. So this is a representation of the developmental trajectory, and this is similar to the previous calculated by other softwares. And then finally, we also calculate the post-studio time based on this topological representation and try to fit it with the actual time point. We got the result as there is a significant p-value with the R value 0.37, and in this part we can conclude that the topological analysis can capture the patterns of the traditional developmental, but there are still a room for improvement, and we believe that further work on data engineering may lead to a better construction of the topological representation, and maybe we can fit the real time point better. And below is our conclusion for this study. Most important one is our findings emphasize the importance of accurately identifying stem cells and highlight the MSC as a preferred method for precise trajectory inference in PhD developmental studies. And these are all the references used in our study. Thank you for listening.